Okay, students, switch on your videos. Let's start today's class. Alright, so today we are going to discuss about what light reaction is. Now, if you people may remember, I told you about light reaction and dark reaction. When and then I said that the light reaction are those reactions which happen directly under the sunlight, isn't it? They help like you can say, they help the plant to produce ATP, NADPH and chemical intermediate, you can say molecules. And talking about the dark reaction, so that reaction is not directly light dependent, but they are dependent on the products of the light reaction, that is ATP and NAD. Okay. Now talking about the light reaction, let us see what all things happen during this. The first thing, the most important thing that happens is the light absorption. And this is done by the chlorophyll molecule, because we know that the chlorophyll molecule has that ability wherein it can absorb the sunlight. Now, after absorbing the light energy, it gets converted, or you can say it converts the light energy to chemical energy, because light energy cannot be used as such. At the same time, water splitting takes place, meaning the water molecule, it splits into hydrogen and oxygen components. Oxygen that is formed by the splitting of water, it is released, right? Because that is the oxygen that we use for breathing purpose. And lastly, the most important thing that happens during the light reaction is the formation of ATP and NADPH. ATP and NADPH, you people already know about ATP. What is the full form of ATP?
Adenosine triphosphate.
Adenosine triphosphate. So ATP and NADPH, they are formed and they contain high energy, and these are chemical intermediates. Meaning whenever the plant cell or the plant process would require any energy, it will come from the breakdown of these chemical intermediates. So the introductory part of light reaction is clear to you all?
Yes.
Yes. Now see, for the process of light reaction to take place, there is a system in the plants which we call as the light harvesting complex. Light harvesting, you understand? The complex that is responsible for collecting the light for photosynthesis, because this is extremely important. Now the light harvesting complexes, they are of two types, photosystem 1 and photosystem 2. We also call them as PS1, PS2. Now remember that chlorophyll A molecule, if you may remember that I said that this is the chief pigment, isn't it? Why is it the chief pigment, students? Tell me why chlorophyll A is the chief pigment and not any other chlorophyll, B, C, or xanthophyll, carotenoids. Why are they not chief pigment? Why is only chlorophyll A the chief pigment?
Because chlorophyll A is there in in all plants and
Chlorophyll A is present, and this chlorophyll A absorbs maximum light. Do you people remember in which region of light does photosynthesis take place? Which color?
blue and
red. Blue and red region. Very good. All right, now talking about the reaction center. Let us see what is the difference between the photosystem 1 and photosystem 2. Now talking about the photosystem 1, so the reaction center, like the chlorophyll A molecule of photosystem 1, they have an absorption at 700 nanometer, meaning they will get activated only and only 700 nanometer wavelength of light is incident on them. Then they will get, like you can say, activated. Before this, they will not go. Talking about the PS2 here, the reaction center has 680 nanometer. Meaning once the light is coming, if it is 680 nanometer, quickly PS2 will come into play, but the PS1 will not. PS1 and PS2 is clear to you all?
Yes, ma'am.
Now see, this is how the whole entire mechanism happens. What happens is that the light harvesting complex, it has all the pigments that are responsible for trapping the sunlight. That is why it is called as light harvesting. You people might have people might remember about rainwater harvesting. So just like that, light harvesting is also there. Now in the light harvesting complex, there are certain pigments molecules, and when they absorb the light, they become excited. Okay, excited means they don't dance and jump along, but excited in the terms of chemistry. You people remember relaxed state, excited state, they jump into another orbit, all of those things you might have studied. So now
Yes.
they release the electron. This electron gets released and it is caught by the chlorophyll A reaction center. Talking about the PS2, like I told you, it works at the 680 nanometer range of light. So any doubts in the photosystem?
No, ma'am.
The whole entire thing is clear to everyone?
Yes.
Okay, Sabiha, am I audible?
Yes, ma'am.
Switch on your video. Okay, tell me one thing, students. Whenever, if someone would ask you, the oxygen that is released as a byproduct of photosynthesis, from where does it come? Where does that oxygen molecule comes from? Hmm? Tell me, you people are saying that everything is clear, but now you're not answering simple questions.
From the water, ma'am.
H2O, there's enough water.
From the water.
Yeah, so what is happening? Yes, you are right, water. But what is happening to the water that it is coming?
It splits.
Hmm. Water
The
molecule
plant,
splits.
the
And
plant
that is the
absorbs
reason
the
why,
water.
yes,
The plant absorbs the water.
no. See, during this particular stage, like I told you, the oxygen is released from the water molecule only. But how? The water molecule splits into two parts, that is hydrogen and oxygen. And then that oxygen, since it is a gas, you can't accumulate it inside the cell. So it gets liberated out through the stomata. Clear? Now the next thing that we are going to discuss is electron transport system. But before that, I want to show you all the differences between PS1 and PS2. Just one second. Is there anyone who's having any difficulty, students? I'll show you that particular picture. Just one second, students. Just one second, I'll just show you. Okay, are you able to see the screen, student? Hmm?
Yes, ma'am.
Difference table is clearly visible to everyone.
Yes, ma'am.
Hmm. See, this is the major differences that are there in between the PS1 or and PS2. Now, when we talk about the PS1 here, P700, 700 nanometer is the wavelength that it gets reacted, or you can say it gets activated at that wavelength. Talking about the PS2, it is 680 nanometer. That is the reaction center. Now, in terms of location, so the PS1, it is lying outer surface of the thylakoid. You know, you people remember thylakoids? What are thylakoids? Tell me. Thylakoids are the parts of the
Yeah.
chloroplast, isn't it?
Yes.
And yeah, so this PS1, it is present on the outer surface of the thylakoid, whereas the PS2 is present on the inner surface. This is another difference. Now the photosystem 1, it is present in both grana and stroma lamellae. Grana means the stack of thylakoids. So in that also it is present, as well as it is present in the stroma lamellae, meaning the part that connects the two grana together. But the photosystem 2, this one is found only and only in the grana lamellae and not in the stroma lamellae. The photosystem 1, it participates in both cyclic as well as non-cyclic flow of electrons. We will study both of them, don't worry about that. Then talking about the PS2, it only and only works at the non-cyclic flow. Photosystem 1 is very versatile. It will work at any condition, but PS2 is a little, you can say, picky. Not associated with splitting of water. During the PS1, the water molecule does not split, but during PS2, water molecule splits and ATP is also formed and oxygen is also released. Is it clear, students?
Yes.
All right, okay. Now you people note down till here, then we move forward. Was there anything left from the previous lecture that you people had to note down? No? All right, now you people can note this.
Done, ma'am.
Everyone's done?
No, ma'am.
Okay, waiting. Done, everyone?
Yes, ma'am.
Done, students?
Done, ma'am.
This difference table is very important. So everyone write these things down, make a column for photo system 1, photo system 2, and then write these points. Okay, that's very important. Yes, students, are you done?
1 minute.
Okay. Take your time, it's all right.
Done now.
Everyone's done? All of you are done, students? Yes or no?
Yes, ma'am.
Okay. Now the next thing that we are going to discuss is the electron transport system. Now remember this whole entire process of photosynthesis. It works on the electron transport, or you can say the electron moves from one place to another, thereby creating energy at every way that it enters. Now, electron transport is a series of electron carriers over which the electron passes. And while the electron passes on the electron carrier, they release energy at every single step. So that is electron transport system. Now comes photophosphorylation. You people know that photo refers to light. Now see, the formation of ATP in the presence of light is what we call as photophosphorylation. Why? Because the word phosphorylation refers to the formation of ATP molecule and photo is the use of light for it. So the process of formation of ATP from ADP and inorganic phosphate using light energy is called as photophosphorylation. Now, all of you know that if we split the ATP molecule, we get one ADP and one of the phosphate gets released. This process is of energy utilization. Whenever energy is utilized, then ATP breakdown takes place. But during the formation of ATP, that ADP molecule associates with a phosphate molecule and ATP is like ATP is adenosine triphosphate, ADP is diphosphate. Clear to you all?
Yes, ma'am. Yes, ma'am.
All right. Now, the ETS, that is electron transport system, and the photophosphorylation is very linked because while the ATP is being synthesized, at that point of time, electrons are also being released. So, from the photophosphorylation side, the electron can move towards the ETS. So, that is why they are very linked together. Now, talking about the types of photophosphorylation, I've written here photophosphorylation. It's photophosphorylation. So photophosphorylation is of two types, non-cyclic and cyclic. Students, do you know what is the meaning of cyclic? It's not cycle. What is cyclic?
When something returns to the origin.
Meaning if you start from one point and then come back again at that one point, that is cyclic, isn't it? Exactly. So this is Exactly, that's cyclic, meaning the mechanism is cyclic, but non-cyclic means it will not come back to its original position. Now talking about the non-cyclic photophosphorylation, what happens here is that this kind of photophosphorylation, meaning the non-cyclic ATP formation or ATP synthesis, it involves the PS2 and PS1, meaning the photosystem 2 and the photosystem 1. Now remember that the photosystem 2 will be acting first and the photosystem 1 will be acting after the photosystem 2. Okay, it doesn't matter if one is given to PS1 and all. First photosystem that will work will be PS2 and then PS1 will work. These two photosystems, they work in series. First the PS2 work and then the PS1 work. Now, the two photosystems, they are connected through an electron transport chain, meaning one like PS2 is connected to the PS1. There is a bridge in between them, and that is the electron transport chain. Now, during the process of non-cyclic photophosphorylation, ATP and NADPH, both of them are synthesized. Now, see, this is what happens. Everyone should practice this diagram. Okay, this can directly come in the examination. So I want you all to understand what the diagram is and what does it tell us. See, what happens is that whenever the light is coming from the sun, it falls on the light harvesting complex. Okay, light harvesting complex of photosystem 2 has what kind of nanometer light?
680.
680 nanometer. So once the light falls on the light harvesting complex, 680 nanometer of light, that gets absorbed by the molecule and it generates electrons, meaning the electrons, they get excited and the electrons jump. Now, when the electrons jump, they are collected, or you can say they are accepted by the electron acceptor. Then the electron acceptor sends it to the ETS. Then from the ETS, it comes to the light harvesting complex of photosystem 1 that has 700 nanometer wavelength required. So once the light reaches there, or you can say the electron reaches the light harvesting complex of the photosystem 1, again the electron will be excited. It will jump and be accepted by the electron acceptor. And lastly, at the end of this whole entire procedure, NADPH and NADP plus will be formed. So this is the non-cyclic. Do you see, if it starts from like the light harvesting complex of photosystem 2, it is ending in a different area and it's not coming back to its start point. So this kind of photophosphorylation will be non-cyclic because it's not in a cyclic form. Now, if you just rotate this picture in your mind, what alphabet do you see if you rotate it clockwise?
Z.
Z. This whole entire process of non-cyclic photophosphorylation is also called as Z key. Z key. Clear? Now, first in the photosystem 2, this is the theory of what I've just said. So first in the photosystem 2, the P680 molecule absorbs the 680 nanometer wavelength of red light. This causes the electron to become very excited and they jump. And whenever they are jumping, they are accepted by the electron acceptor. And then they are sent to the ETS. This movement is downhill. Always remember, when it's going up, that movement is uphill. When it's coming down, then that is downhill movement. So this particular movement is the downhill movement or redox potential scale. Now, once the electrons work in the photosystem, electron transport system is done, Now the electrons, they are sent to the light harvesting complex of the photosystem 1. Now the P700 gets excited. Again, the jumping of the electron takes place. Ultimately, the electron is accepted by the electron acceptor. And then NADPH, NADP plus, ATP, all of these molecules are formed. Now the electrons are then moved downhill again to NADP plus, meaning whenever electron will be added to NADP plus, it will form NADPH. Also, the addition of electron to the NADP plus reduces it and forms NADPH. So non-cyclic photophosphorylation, is it clear to you all? Is there anyone who has any doubts? First of all of you switch on your videos. Only one student has, and the rest of you, where are you? Are you there in the class or not? Very good. Now, this whole scheme of transfer of electrons from PS2 uphill to the electron acceptor, downhill to the EPS, and then again to the PS1 uphill. Right, this is a Z scheme because it's actually it looks like the alphabet Z. Is it clear, students?
Yes, ma'am.
Now, if you remember, while you were writing the differences between PS2 and PS1, you might have written that the splitting of water molecule takes place in the PS2. So now we are going to talk about how the water molecule split, why it splits actually. Now the electrons removed from the PS2, they must be replaced, isn't it? Because if it is a procedure, it has to keep on constantly going. So electron replacement is also required. So once the electrons are removed from the PS2, they must be replaced. And this replacement is done by splitting of water. Meaning, when the water molecule splits, it gives us 4 molecules of electrons. And this electron is used in the process wherein after PS2, meaning in PS1. Now, the splitting of water is associated with the PS2 as it replenishes the electron. Are you people getting where the electrons have to be replaced? Tell me in this diagram where the electrons would have to be replaced. The thing that I'm teaching you right now. Tell me. You can use like
after it gets transported to LHC or photosystem 1.
You're not audible, dear. See, so during this process, what happens is that the PS2, it has, you can say, it is present in the membrane of the thylakoid. We have studied that. And it that replenishment, like I'm saying, the replacement, meaning once the electron has jumped from the light harvesting complex, it means that now the light harvesting complex does not have enough electrons. So that replenishment or refilling of electrons is done by the splitting of water. Is it clear to you all?
Yes, ma'am.
Then comes cyclic photophosphorylation. Now this method will be a cyclic method, right? Starting from one point, ending at that one point. And in the cyclic photophosphorylation, only and only PS1 is active. There's no use, there's no need of PS2 in this condition. This process takes place in the stroma lamellae. Now what happens? Whenever the light energy is incident on the P700, the electrons get excited, they jump and they are accepted by the electron acceptor. Now after that, they are sent to the electron transport system. That is for Continuation. Do you see how the cycle has to be continuated? So this is how the cyclic photophosphorylation takes place. What happens here? The P700 accepts the 700 nanometer wavelength. The electrons of the PS1, that is photosystem 1, it gets excited and the electrons, they start jumping. Now these electrons are ultimately accepted by the electron acceptor, and then it is sent to the ETS, and which sends it back to the light harvesting complex. So it starts from one point and it ends at that same point. So that is cyclic photophosphorylation. Is it clear, students?
Yes, ma'am.
Now these are the differences that you people have already written. Now you can note it down. Also, these diagrams are very important. Everyone will make the diagram.
Done, ma'am.
Everyone's done?
Yes, ma'am.
Yes, ma'am.
Done, ma'am.
Just a minute. Done, ma'am. Done, ma'am.
Everyone's done?
Yes, ma'am.
Okay, everyone draw this and practice it because it can be asked. Or if you draw this in the support of your answer, you will get full marks.
Done, ma'am.
Everyone's done?
Yes,
Students, did you label the diagram properly? Like which part is photosystem 2 and which part is photosystem 1? Did you label it nicely?
yes, ma'am.
Okay.
Ma'am, can you please scroll down?
Yes. Is everyone done?
No, ma'am.
Okay. Just wait a few minutes, Shobita. Okay.
Okay.
Done, ma'am. Done, ma'am.
Everyone's done?
No, ma'am.
Okay.
Done, ma'am.
Done, ma'am.
All of you are done, students?
No, ma'am.
Okay.
Done, ma'am. Done, ma'am.
Everyone's done?
Yes, ma'am. Done, ma'am. Done, ma'am.
Done, everyone?
Then
Done, students?
Yes, ma'am.
All right. Okay, students. We'll keep it till here only for today's class.